Hello everyone, you are watching Bioinformatics Insights and my name is Ashfaq Ahmed and I work in the field of structure biology and bioinformatics. Today we will cover part 6 of the computer-aided drug designing. Before part 6 we have covered part 1 to part 5. Among these 5 parts, we worked on how to identify a receptor in UniProt and ChEMBL. We also worked how to download the required data in the form of bioassays from ChEMBL. Then we dealt with some data management and data filtration by rule of 5 or Lipinski rule. And finally, we also worked and clean our data from some unwanted substructures, or we can also call them PANs. And then we analyzed the data based on the fingerprints. In part 5, we cluster the compounds based on the structural similarities in larger and smaller clusters, and we use some different variables and different algorithms of Dynamotus and others. So if you haven't watched these 5 videos and if you are watching this part 6, I'm not sure you will understand this or not. Therefore, I suggest you to first watch from part 1 to part 5. In today's video, we will learn how to find common substructure in the available data. In the field of chemoinformatics, it is also called MCS, maximum common substructure. This is almost the last lecture about this series. Next we will move to some machine learning and other stuff, and hopefully we will cover this course effectively and we will learn too many things. Before to proceed, I just want to explain one thing. Most of the time I'm receiving different messages through email or through WhatsApp or other social websites, and they are asking like, is it necessary to clean our data from the unwanted structures or the unwanted substructures? So I want to answer here again. If you have some question, you can ask, but the answer is it is not necessary. But if you want, you can. I also received a question that when I do this kind of filtration, the amount of data which is left with me becomes very minimum or minimal data. So what should I do? So I straightforwardly I replied him that you can skip this step. The purpose of these steps that you should know that such strategies can be done. Although these strategies are not hard and fast rules, so it's up to you which strategy to adopt and which strategy to skip. Okay, so let's move to part 6. We will also cover some basic introduction and other stuff. Okay, so how to access? I'm sure you will have some idea now. Google or Colab research, then go to GitHub H100, then click. By the way, I dropped this link in the description section so you can copy from there. BioPython. Here you need to click Create Project, and then here you need to open this one Max by clicking here. I have already opened and connected. I have already connected this tutorial, so therefore, but to open it you need to click here. You will have this window. Now you will click here the connect button, and your Colab notebook will be connected. As usual, the first part contains my information and the link for this YouTube channel, and this is the information of the authors who worked on this notebook initially and who prepared this notebook. So if you want, you can also search them or visit to their websites. Okay, what is the aim of this tutorial, or what is the aim of this part 6? It is basically a connection of part 5. In part 5 we cluster our data, we cluster our structures in particular in different clusters, in different groups. Here we will find out the common structure, the common substructure which is present in those clusters. It will help us in future settings. So this is the aim of this talk, to find out a common substructure. And what we will work on, so we will work on the FMCS algorithm. I have dropped a link here below, so if you want to read, you can read about this algorithm. We will use this algorithm in RDKit, and we will find out the common substructure, or the MCS, or the maximum common substructure. So content in the practicals will be, we will load and draw some molecule. We will run the FMCS algorithm with different input parameters. We will discuss in this course, even in today's session, and we will find out some diverse set of EGFR compounds downloaded from ChEMBL. You should know that in this course I am dealing with EGFR compounds, so therefore I am going or I am stick to that EGFR. So, and we will identify some MCS or common substructure with some different cutoffs or some different parameters, and these are the references. If you want to read some paper, and in fact I suggest everyone to read different papers and to read different literature. In reality, only this course is not sufficient. You need to have much more information. Okay, so let's come to the theory part, that what actually is maximum common substructure. So these are actually molecules, or a part of a molecule. If you see these 4 structures, you can see some area are mentioned in bold. That is this one here in this structure. This area is mentioned in some pink shading and bold representation. Again some pink shading and bold representation, also some pink shading and bold, and as it is in the 4th structure. What it means? It means that these 4 structures share this bold area or this pink shaded area, and if you look at them, then all these 4 structures share this area. So in other words, we call it, this will be the MCS of these 4 structures. So from this example is based on only on these 4 structures, but in reality we will find out such kind of MCS dealing a huge amount of data, maybe in thousands, maybe more than that, or maybe in hundreds. So therefore, for understanding point of view, this will be the the MCS, or the maximum common substructure. Okay, this is some information which I discussed, and here if you click here, so it will lead you to the interpretation or the details of the FMCS algorithm which we will use. Okay, but this course is not about to teach you the MCS algorithm, so I'm not going in that detail, but for sure I will tell you that what this algorithm will do. For example, this is your structure. So this algorithm breaks your structure into smaller pieces or into a smaller fragments, like this you can see, or like this, and then they compare those pieces to the whole data set, and then they rejoins, and they will tell you that which part is common in the maximum portion of the data. Okay, so this is all the critical or the minute details, so if you want to read you can. Okay, now we are going to start and to start our analysis, and here you have to run this is the Conda installation, and once you run it, remember you will see a restart here, but I have already run it, so therefore I did not see a restart. Okay, the second step is you have to run these codes, and it will take almost 15 to 16 or 17 minutes. So it's better that once you run this, you can go outside and have a cup of tea or coffee, so it will take some time. Okay, then we will import, and after this import job we have import certain things from the RTKit, from the matplotlib, from numpy, panda, pathlib, etc, etc. Okay, now here we will load the data generated in part 5. What we have generated in part 5, I will look to you. This is the part 5 compound clustering, and if you remember, or the data must have in your computer if you have run part 5. In code 18 we had run some analysis in code 17, but in code 18 what we have done, we have saved molecules from the largest cluster, so that in other tutorials we can use it. It is the heading, it was the heading, and the name of the data set we have saved, molecular set largest cluster SDF. So here in this talk tutorial, what we will do, we will just click here, we will just upload, and we will here is the data, we will upload the data here. Already I had this data, I have uploaded here, it's here. Now what you will do, click here, copy path, and then Control V, it will paste the copy or the path here for that file. Now you can run this. It says that the dataset contains 157 molecules loaded. It means 157 structures. Okay, now the second cell says show only first 10 molecule. I have run it, and now you can count. So these are the top, not top 10, actually the first 10 molecules among which is present in this data set of 157 compounds. So if you want to make it 5, you need to just change this number to 5. Otherwise, or if you want it more, so you can. So it is just a visualization of 10 structures, nothing else. Okay. Now we will run the FMCS algorithm with different parameters. So first of all, in the simplest case, we will run it that the MCS1 is we are making a new variable, and we will run it, and it has calculated that the MCS1 contains 24 atoms and 26 bonds, and these are the information we are not understanding, but the algorithm or the RD kit understands. What it has done, simply it has checked those 157 structures, and they told us that there are 26 atoms and 24 atoms and 26 bonds which are shared by these compounds, these 157 compounds. Okay, now I say the next thing is that we say, okay, draw representative structure from the MCS1, maximum common structure 1, and it said that most this kind of structure is present in your this cluster. Remember what we have uploaded, those 156 7 compounds. Remember from where they came? It came from our largest cluster we have calculated in part 5. So if you want to analyze your smallest cluster or your other cluster generated there, you can do the same thing. You can upload that data here and it will calculate. Okay, now we will define some helper function to draw the molecules and with the highlighted MCS. What it means, the highlighted MCS, in the start I showed you with some bold and pink color, so that is actually the highlighted MCS. Okay, it's done. Now we will print top 5 molecule. It's already printed because before making this video I had run this, so let's rerun it. Okay, now if you see them clearly or with some attention, so this part, or you just see here this part, the double 6-member ring and connected by N nitrogen and the single 6-member ring, they are almost present in every structure. You see here is the extra ring, but this thing is present almost in all the structure. Now I want to print the 10 structure. Let's see. Now these are the top 10. Okay, so this way you can change how many structure you want to print anyway. Okay, now you can also save this data as an image and that will be the SVG image. But if you are not easy with SVG, so I should tell you that SVG produce high quality figures, and there are numerous number of programs by which you can convert the SVG figure or the SVG materials to your PNG, JPG, TIFF or PDF, etc., etc. So once you click it, you will have this kind of figure and it will be saved here. Okay, so from here you can download if you want them in future. Now that was just a default. Here I told you that it is just the simplest running of the MCS algorithm. Now we will use some threshold. Okay, what kind of threshold we are using here? So I'm using the threshold of 80%. Now I will ask the program to calculate a structure or MCS for me that is present in 80% of the structure. Okay, so you can see here threshold 0.8. If you want to make it more strict, let's say 90%, then you can change this 0.8 to 0.9. Or if you want to make it more relaxed, let's say 60%, then you can do it. Let's say I am using it with 60%. That's it. It is up to you, and there is no hard and fast rules. Every point has its own pros and cons. Okay. Welcome back. It took almost 2 minutes, and I had paused this video to save your time. Okay, it has generated some output. It says that 30 atoms and 32 bonds are in common, and that is present in the 60% of the data. Okay, now I will click here, and why it says 0.8%? Because I did not change it to 60%. Okay, so this is the MCS1 or the MCS2. Okay, and because I asked them to give me MCS1 and MCS2 like this, and if you see, it's the similar thing. They gave us the similar kind of structure, double rings and a single ring like this, because it's the same data. But why I rerun it? If you carefully look to this code, here we have introduced a new parameter of threshold. Okay, so in the beginning we did not ask for such kind of threshold. Now let's print top 5. These are the top 5 molecules. All of them contain this substructure, you can see. Now here we are using we are learning to use some more strict parameter. For example, most of the time we do not want to lose our rings. What are rings? Rings are actually these 6-member rings are the aromatic structures. So most of the time people or the scientists or researchers, they do not want to lose their rings. So therefore we will use an extra parameter, ring match region only true. It means that we are asking the algorithm to find out the MCS that satisfies 60% of the data. I will change it to 60% because the In prior step I used this 0.6, and we also asked to ring match, do not lose the ring region. So let's see. It will again take almost 2 minutes, so I will pause this session. Okay, we'll come back. It took almost more than 1 minute, and now we will see those structures. And it tells you that in the ring match MCS3, this region actually I am matching. So you can see here they are matching. Why it's still printing 0.8? Because I did not change it here. It's a legend is here. This is the legend information. Okay, now I tell you an interesting thing. I just teach you how you can do such kind of thing, but here the FMCS module is present with the RD kit. So if you want to do more stuff, you can click here and okay, leave it. Okay, so this is here you can read and you can apply. After reading you will learn how to apply much more strict options. Here we have applied two options, the ring match and a threshold. But if you want to consider some specific atom and some specific bond or some specific valence matching, then you can do that. For example, I will give you an interesting example. I was working with the protein NDM1, and NDM1 is crystallized with the hypertensive or cardiovascular drug that is called captopril. The captopril contain cysteine, or I would say the sulfur, not cysteine. The captopril contain sulfur as a side chain, and my job was to find out a drug that contain a cysteine, because from captopril attachment we came to know that NDM1 active site is pretty much sensitive or pretty much prone or easy to bind with a compound having sulfur. So here I applied different strategies and I strict myself to consider the maximum common substructure or the MCS having a specific atom like sulfur, or maybe you are interested with some specific valence number. So that's it, you can do it. You can read this document here and you can apply some much more things in your research. The basic aim of this course is to give you a stepping stone or a foundation, and I hope some of you will go beyond and further. Okay, now this is very interesting. In part 1 we had some data. If you remember, we downloaded some data before the filtration and then we converted our IC50 to PIC50. Now we will use that data here. The data name at that time I save it with RO5 properties 1. This is the name I choose it. I don't know what is the data name in your case. So what you need to do, just find out the data generated from part 1 and upload it here the same way upload, and it is here. I have already uploaded. Now then copy path and then paste it here. Okay, now, okay, what we have asked to the program that only consider the compounds whose PIC 50 is greater than 9. Maybe in your case you have not so much data, and in your case the active molecules or the active part starts from the 7 or 8, so then you need to change this 9 to 8. But here we are using PIC 59. So we want to find out the MCS based on the whole data whose PIC 50 is greater than 9, or that data which is active in a bioassays performed or present in the ChEMBL. So I load the data and that says the total number of compound is 6816. Number of compound with 322 compounds are actually those which have the PIC 59. So here we have only selected those compounds. Okay, and then we ask the program just this maybe some, let me check this error. Okay, the error is some about the panda tool, so probably this notebook is connected since long ago, and I'm not going to run this again. If you had this problem, I'm sure you will not have, but in case if you have, but then in that case you need to rerun this step. If I run it, I have to pause this session for 20 more minutes, so it is not a very critical error. So first of all, it will not be generated with you, but still if it is generated, so the answer is that you reinstall that installation script, it will be solved. Or if it is not, so I hope we will have no problem in further in running the other cells. Okay, now we mold diverse samples 50. Okay, let me run it first, then I will tell you what we did, so that I will be sure that okay. So what we did, we uploaded our data. 322 compounds were found having greater than 9 PIC 50. Here we were interested to see 3 compounds from the list of 322, but because of the panda tool error we remain unable, but that is fine. The other codes are still okay. Now we just ran these cells to find out the MCS in these 322, and we asked the program just to randomly pick 50 molecules from this set and then calculate the MCS. If you want to pick all of them, you mention 322, but it may take a bit of time, so that's okay. Here we choose the threshold 0.7, or like we did before 0.8, 0.6. You can also use the ring structure matching, and here they will tell you that you have this kind of structure, the ring match. There is no proper ring match, but this kind of structure is a common substructure. And if you want to highlight them, so it will be like this. Okay, and if you see, it also tells you the same substructure here. And then here we have used some different cutups, and like we selected some percentage, and then we use some cutups, and here is the percentage value. Let's say this structure is present almost in 100%. Okay, I choose it value 70. Let me check. This structure is present in almost 100% of the compound, or this shape, or these bonds. Or what about 40? Okay, so after running this code, we will formally, okay, in 40% of the structure, it is satisfied. Now a question may comes in your mind that this structure was pretty much common in the largest cluster we were dealing in part 1 today, in earlier parts. But once we load our own data, the EGFR, so we see this cluster in 40%, but in 100% we see some other structure. The reason is, in the start we were dealing the data, the 156 molecules, only from the largest cluster. Here we are dealing 322 molecules that contain some of the molecules, some of the representation from the largest cluster, and some from the other clusters. So that's the difference. Okay, I hope you have learned something, you enjoyed this part, and if you have any question you can ask. I will try my best to answer. Still I have some questions remaining, but nowadays I'm dealing with some busy schedule, so that's why this video is coming I think after 20 days. So I will try my best to cover this course as quickly as possible, because on the other side I have much more work to share with you, much more diverse videos to share with you. But I don't want to leave this course in the middle and start uploading some other things. So therefore I have refrained myself that, like, first to complete this course, then I will share other stuffs or other videos with you. Keep supporting this channel. If you have not subscribed yet, please subscribe, and stay happy, and God bless you. Bye bye.